Well, the next topic in this chapter is immunoassays and ophthalmos. Let's consider the immunoassays first. Okay, an important application of fluorescence is in immunoassays, which employ antibodies to detect analyte. In this case, analyte is antigen. Okay, what is antibody? Okay. Immunoassay utilize the antibody and antigen interactions. Okay, antibody is a protein produced by the immune system of an animal in response to a foreign molecule called an antigen. Okay, our body or animal body has immune system to protect the bodies. Okay, and then in the process of immune system, antibody is produced, okay, to um, detect, uh, to what detect the antigen. Okay, antigen is a foreign molecule, right? And is a very harmful foreign molecule. Okay, the antibody recognizes the antigen that stimulates the synthesis of the antibody. Okay, the formation constant for the antibody antigen complex is very large, actually it's extremely large, whereas the binding of the antibody to other molecules is weak. Okay, yeah. So now you want to detect a specific compound. Okay, and then you inject the compound into an animal. Usually people use a rabbit. Okay, and rabbit after injected the uh, uh, the specific compound, the rabbit immune system now synthesize antibody to specifically bind the antigen. Okay, so this antibody and antigen interaction or reaction is very specific. Okay, but remember that this antibody doesn't have any catalytic properties. Okay, but in the meantime, I think you are aware of enzyme and substrate reaction. Okay, a specific enzyme catalyze specific substrate. Okay, so enzyme substrate reaction is also selective and highly selective, but this selectivity of this reaction is less than the antibody and antigen interaction. Okay, but one of the advantages of enzyme is their catalytic properties. Okay, for example, the one enzyme may be uh, catalyze a lot of substrates molecules. Okay, for example, the glucose oxidase. Yeah? Glucose oxidase in our blood system, uh, it one glucose oxidase enzyme oxidize a lot of maybe more than 10,000 what 100,000 glucose molecules. Okay, but antibody, although it is very specific to antigen, but it doesn't have any catalytic um, property. Yeah, so immunoassay is being uh, widely employed. Okay, so this woman, the Rosalind Yallop, she is a physicist. She won the Nobel Prize in Medicine in 1977. Okay, she developed the immunoassay technique using proteins labeled with radioactive iodine. Okay, this iodine, mass number 131. This is radioactive. Okay, so this immunoassay is widely used. Right. Let's consider here the a specific technique called ELISA. Okay, it sounds like a very pretty woman's name. ELISA stands for Enzyme Linked Immunosolvent Assay, and this technique is also very sensitive to specific analyte. Okay, now your purpose is to detect analyte concentration. Okay, let's say this analyte is a protein. All right, and the concentration is extremely low, maybe ppm or ppb or even ppt, okay, part per trillion, right? How do we detect such a low concentration on a light? Okay, ELISA gives you an answer. Okay, well, this is the process of ELISA preparation. First, you prepare the polymer support, and on the surface of the support, you fix, okay, the antibody one. Okay? Usually, antibody shape is represented by this Y. Okay, so you now, absorb a lot of antibody molecules on the support surface. Okay, and then you add sample containing analyte, but very unfortunately this sample contains not only analyte but also many other molecules. Okay, and analyte concentration is extremely low. Okay, your purpose is to specifically detect, determine this analyte concentration. Okay, then after preparing this antibody 1 surface, and then now you add the solution and you wash, okay, carefully wash, yeah, to remove unbound molecules. So this analyte, this is now antigen. Okay. Mm. Since it has uh, interaction with this antibody 1, this specific interaction. So after incubation and wash, you have only the analyte bound to antibody 1. Okay, so interaction between these two molecules is specific. But since your analyte concentration is quite low and there are a lot of antibody 1 molecules on the surface, so some antibody 1 now uh, bound to analyte protein, but some antibody 1 is empty. Okay, because the analyte, uh, the molecule, the its number is less than the antibody 1. Okay, and then what? And then you go on to add enzyme labeled antibody 2. Now you prepare this antibody, and at the end of this antibody you linked. Okay, that's why it says enzyme linked. You linked enzyme. Okay, this enzyme is covalently bound to antibody 2. Okay, and very interestingly, this antibody 2 binds this unrelated protein, but on the other side. Okay, the uh, protein is a very large molecule. Okay, it's very large, and on one side is bound to antibody 1, but the other side is bound to antibody 2, which is covalently bound to enzyme. Okay, this enzyme. And then what? There are two modes of detection. One mode is to uh, you use colorless reactant, but this reactant is um, what undergoes reaction through this enzyme, okay, which is bound to antibody 2, and this reactant actually initially is colorless, but after after being reacted by this enzyme, it becomes a colored product. Okay, so you from the color change, or you can say, oh, there is an analyte. Okay, yeah, this way people developed a placenta sensor which indicates very easily whether the woman is pregnant or not. Okay, another way of detection is to uh, use non-fluorescent reactant. Okay, but this non-fluorescent reactant now becomes fluorescent product after being reacted by this enzyme. Okay, so here this the thickness of color depends on the number of analyte, okay, and also the fluorescent intensity also proportional to the number of analyte, okay. So by detecting, okay, this fluorescence or by measuring the intensity of color, you can uh, determine the analyte protein concentration. Yeah? This is the principle of ELISA. Okay. So this immunoassay is very sensitive. Okay. So it has been applied to environmental analysis. Okay. So commercial immunoassay kits are available. Okay, for screening and analysis of pesticides, industrial chemicals, explosives, and microbial toxins at the parts per trillion. Okay, this is extremely low concentration. 2 parts per million, still very low levels in groundwater, soil, and food. Okay, you see the pesticides, yeah, and the chemical explosives and toxins, yeah, and those compound level in environment, yeah, is not high, it's quite low, but at any rate you need to determine uh, the concentrations of these the molecules, okay, and then immunoassay may be the answer. An advantage of screening in the field is that uncontaminated regions that require no further attention are readily identified. Okay, so in the when you apply this technique to environmental analysis, you have a advantage of this immunoassay. Okay, uh, that uncontaminated region. Okay, which requires no further attention, okay, readily identified, okay. So you can say that, oh, this area is uncontaminated and that area is contaminated, okay. An immunoassay can be 20 to 40 times less expensive than a chromatographic analysis. Okay, for the environment analysis, immunoassay is not the only solution, but there is another solution, which is chromatographic analysis. Okay, but this chromatographic analysis is also very sensitive and very important. I will give you lectures later, but the immunoassay has some advantages, like the price of immunoassay is 20 to 40 times less expensive, okay, and it takes a very short time. The photo chromatographic analysis takes a long time, but immunoassay can be completed in zero point 0.3 to 3 hours in the field. Okay, this is on-site analysis using 1 milliliter samples, and sample size is quite small. In the meantime, chromatography generally must be done in the lab. Okay, because you cannot take your chromatography instrument into the field. Okay, so you have to take the sample in the lab, and it might require several days for the preparation for pre-treatment, something like that, because analyte must be first be extracted or concentrated from little quantity. Okay, here immunoassay only you need milliliter scale, but for chromatography you need much higher, you know, much larger samples, maybe liter scale, okay, to obtain a sufficient concentration. Okay, but immunoassay also has a disadvantage. One of them is it is very difficult to get antibody. Okay, the getting antibody is not a simple matter. And now let's consider time resolved fluorescence immunoassays. Okay, this immunoassay can be applied to the time resolved fluorescence, okay, measurement. When do we need this specific technique? In this technique, you may need a laser beam, but this technique is useful when background fluorescence is strong. Okay, the sensitivity of fluorescence immunoassays can be enhanced by a factor of 10, okay, maybe down to 10 to minus 13 molar analyte, okay. This is what, 0.1 picomolar, right? Extremely low concentration, 100 femtomolar concentration, okay. This extremely low concentration can be, very accurately determined by time resolved fluorescence technique, immunoassay technique, okay. The one example is to determine the lanthanide ion europium, okay, this one, by by measuring the luminescence, here in this case the fluorescence, the luminescence measurement, okay. Organic chromophores such as fluorescein are plagued by background fluorescence at 350 to 600 nanometers from solvent, solute, and particles. Okay, which means that, you know um, to apply the fluorescence technique, you may use this fluorescein, which is very strongly fluorescent. Okay, but this fluorescence signal are plagued, disturbed by background fluorescence in this wavelength range. Okay, maybe solvent, solute, or particles may uh, be fluorescent. Okay, so you need more sensitive technique, okay, which is the time resolved fluorescence immunoassay. This background decays to a negligible level within 100 microseconds after excitation. To measure the fluorescence, you need excitation light, okay? Sharp luminescence at 615 nanometers from europium ion, however, has a much longer lifetime, falling to 1 over Te of its initial intensity in approximately 700 microseconds. Yeah? I will explain in detail in the next slide. Time resolved fluorescence measurement, luminescence is measured between 200 and 600 microsecond, I'm sorry, not second, microsecond, after a brief laser pulse at 340 nanometers, okay? So you need laser beam, okay? So this technique is maybe a very expensive, okay? The next pulse is flashed at 1000 microsecond later, and then the cycle is repeated, approximately 1000 times per second, okay? So you can measure one measurement, okay, at 1000 microsecond. All right, this is 1 millisecond. So within 1 second, you can measure 1000 times. Okay? So this is why this time resolved fluorescence immunoassay is very, very sensitive. Rejecting emission within 200 microseconds of the excitation eliminates most of the background fluorescence. Okay, here is the illustration uh, of this technique. You see here. Now, x-axis is the time, measuring time, and uh, this y-axis is the fluorescence intensity. Now you excite, the system with the laser pulse here, okay, very short pulse, and then, there is a fluorescence not only from your molecule, okay, which is your species, which is now europium, ion, but also from, the background, like a solvent, okay, like the particle, okay, all kind of background fluorescence is there. But fortunately, this background fluorescence decays very rapidly, okay. About 200 microseconds after laser excitation, this background fluorescence intensity, almost decays to zero level, okay. But this emission, okay, this fluorescence from europium, this ion, this fluorescence also decays with time, but very slowly, slower than this background signal, okay. So you can measure between 200 to 600 microsecond, okay. This is the window for the fluorescence measurement, okay. Why not? After maybe 800, but here the fluorescence intensity is low. Okay, this is why this time resolved fluorescence immunoassay is very sensitive. Yes, this is actual application. Okay, and this part is the same as ELISA. Okay, but instead of using this enzyme, in this case you bound chelating group here. Okay, chelating group. This is not enzyme. This is chelating group. Chelating group, they binds this europium ion. Okay, and then what? As long as this europium ion is bound to this chelate, there is no very weak luminescent. Okay, but when you lower pH and add soluble chelator, and then this europium ion now is released from this chelating group. Okay, now this europium ion is free, and it is highly luminescent. Okay, so by measuring this luminescent intensity, you can determine the europium ion concentration. Okay, that's the principle of time resolved fluorescence immunoassay. Okay.